When I started my lab, the purpose from a physics perspective was to develop those next set of eyes at the single molecule level. So if you look at what's in clinical trial right now, it's many lipid particles containing RNA therapeutics, also other molecular therapeutics made of DNA and RNA. We needed a way to confine those molecules so we can watch them for a long time, but in cell-like conditions. So be able to look at many single molecules and their interactions, one at a time, untethered so they can explore their conformational or shape distributions. So CLIC stands for convex lens induced confinement. So this provides a way to confine the molecules into, say, open face wells. It lets us get to the nanoscale. Two specific applications that have been the greatest interest are the lipid RNA complexes.
Sabrina's approach is a pretty novel approach, and it really does allow us to be much more precise in our characterization of these lipid nanoparticles, which, as you know, are now being used for many, many applications, vaccines being just one example.
So the kinds of questions we've been invited to collaborate on with developers are how to get maximally loaded lipid with RNA, understanding the relationship between these very microscopic properties experienced by, if you were a lipid particle with RNA, you might have an ideal size to enter the cell, an ideal composition to deliver the RNA safely and efficiently. And so if we can understand those properties very quantitatively, it gives us the tools to then optimize them.
And so it allows you a much more precise and a more rapid analysis of what one has in your solution. So you can see this being used as a quality control process for making these genetic medicines, amongst many other applications, actually.